In this video, I'll review concepts from Chapter 24 of Human Anatomy and Physiology, 11th edition. This chapter focuses on metabolism. We're going to go through a few key concepts. Please note that within this chapter, we are not covering everything, so pay attention to the topics that are covered here in lecture, and that should help guide you through your reading of the chapter. There are three major macronutrients that make up the bulk of ingested food. These include lipids, proteins, and carbohydrates. Metabolism is the chemical reactions of cells that involve nutrients, such as those three macronutrients just listed. There are two metabolic reaction types. We have anabolism and catabolism. So anabolism is building bigger molecules from smaller molecules. An example would be building proteins from amino acids. Catabolism would be the opposite. It's the hydrolysis or the breakdown of larger molecules into smaller. So this would be like breaking down proteins into individual amino acids. Note that digestion and metabolism are going to work together. So there's a lot of overlap there, but they are technically different things. So the GI tract is involved in digestion and absorption. Those breakdown products, the breakdown of these nutrients, proteins, carbohydrates, and fats, would be through metabolic reactions. And then you can see that those metabolic products are going to travel through the body into your tissues, where they are going to be metabolized. So we have catabolic reactions that are used for ATP synthesis. So we're making ATP, as shown down here at the bottom. This is called cellular respiration. We also have anabolic reactions for nutrient storage. So all of this storage of nutrients, such as proteins, glycogen, which is a store product for sugars, and triglycerides, store product for lipids. As I mentioned in the previous slide, that set of catabolic reactions that provide ATP or cellular energy, that's called cellular respiration. So it's the capture of energy in the form of ATP. This requires the passage of electrons from glycolysis and the citric acid cycle to oxidative phosphorylation. And I'll demonstrate each of these in the coming slides. So each of these is a metabolic pathway. So one is glycolysis, another is the citric acid cycle, and a third is oxidative phosphorylation. But all three of these are part of cellular respiration. Cellular respiration involves a series of what are called redox reactions. These redox reactions involve the passing of electrons between molecules. So you'll see this word redox, R-E-D, stands for reduction, and OX stands for oxidation. So let's look at what those two words mean. Oxidation is the loss of electrons. Within the context of metabolism for this class, you can also think of oxidation as the gain of oxygen or the loss of hydrogen by a molecule. Reduction is the opposite. Reduction is the gain of electrons. Or for the purpose of this class, you can think of it as a loss of oxygen, or a gain of hydrogen. So oxidation, loss of electrons. Reduction, gain of electrons. I've heard people say oxidation is loss, so oil rig. So oxidation is loss and reduction is gain. That might be one way to remember it. Oil rig is an abbreviation, an acronym. Oxidation and reduction become paired. So you can think of it as one molecule is losing electrons, while the second molecule in the pair is gaining those electrons. So the electrons are moving from one molecule to another, and that would be redox. That's that pairing of oxidation and reduction. So one molecule is oxidized, and the other molecule in the pair is reduced. So now let's start looking at metabolism. So two molecules that are particularly important for these redox reactions in our metabolism are NAD and FAD. These are called coenzymes because they're going to carry electrons to different parts of the cell. So here you can see that both of these, NAD and FAD, are involved in redox reactions. So when NAD is reduced, so when it gains an electron, it moves from NAD and it becomes NADH. When NADH is oxidized, that means it loses an electron. So you can see here it becomes NAD+. So this cycle is going to happen in multiple locations within the cell, and it's going to contribute to different metabolic pathways. And you can see down here, FAD, it follows somewhat the same pattern. We've got FAD becomes reduced, so it gains an electron. Actually, it gains 2 electrons, becomes FADH2. FADH2 can become oxidized, loses those electrons, and becomes FAD. So that cycle is also going to occur in multiple metabolic pathways. The bottom line here is NAD and FAD are the oxidized forms of these coenzymes. NADH and FADH2 are the reduced forms of those same coenzymes. So it's these cycles that are going to be helping with the metabolic pathways within our bodies. All right, so the big purpose of these redox reactions in metabolism is to create energy, so to synthesize ATP. And there are 2 points at which, or 2 mechanisms by which ATP synthesis is occurring. First, we have substrate level phosphorylation. So this is what's occurring within glycolysis as well as within the citric acid cycle. And so for substrate level phosphorylation, we have an enzyme that is directly moving a phosphate group from a substrate to ADP. So we're adding a phosphate to ADP, which is adenosine diphosphate. And when we add that phosphate to that molecule, it becomes adenosine triphosphate. So there are 3 phosphate groups, ATP. So that's substrate level phosphorylation. Oxidative phosphorylation is where your body is going, or your cells are going to use the electrons from NADH and FADH2. We have this electron transport chain that is transporting and using the energy from those electrons in NADH and FADH2. It's creating this proton gradient, so it's pumping a bunch of protons to form a high concentration of protons in the intermembrane space of the mitochondria. And then that proton gradient helps to power something called an enzyme called ATP synthase. It's a membrane channel, and as the protons flow through, that runs this motor, and that is helping to convert ADP to ATP. And that's called oxidative phosphorylation. So this is occurring in the mitochondria, whereas substrate level is occurring within the mitochondrial matrix as well as in the cytosol. I'll leave you with this question. The conversion of FAD to FADH2 is an example of which of the following?